Welcome dear students. Let me introduce myself. I am Dr. Mayur Patil, working as an assistant professor in botany. And in this video lecture, we will be talking about germplasm conservation and its type. It is present in the subject advanced plant biotechnology. So let's get started with the lecture. So what kind of topic we are going to cover in this lecture? In this lecture, we will talk about germplasm, what exactly it is, then what are the different, what is the concept of germplasm conservation, then different types of germplasm conservation. So let's get started with the lecture. So what is germplasm? If you try to define the germplasm can be defined as a live source of all the genes such as seed or any plant part which can be conserved for long time and can be regenerated whenever required in the future. So germplasm literally means the seeds of the plants or the fetus which is present in the womb of the woman, that can be a germplasm. So what is the importance of germplasm? It contains all the information which is required for the generation of entire future plant. Means it has all the genetic information. It is total blueprint of future generation. Now why there is a need of conservation of germplasm? So if we see now, the genetic diversity of all the living organisms has become at very threatened due to the diversity loss or destruction which is occurring in the nature. So to maintain this diversity, biodiversity, it becomes imperative, it becomes compulsory to conserve the germplasm, because once the diversity get lost, we cannot recover it back. Okay. So what are the different advantages of this germplasm conservation? It produces the gene pool of different genes which act as a reservoirs. Means there are now the concept of hybridization is also there, and in hybridization we cross genetically dissimilar plant to produce the improved or highly improved variety. But at the same time, the land races or the original germplasm needs to be conserved, because if you try to produce all the hybrid varieties, then there will not be any original or pure variety. So for that purpose, it is very important to conserve the germplasm, and it is important in the conservation of rare endangered species as well as commercially important species, because rare plants, or you can say endangered, according to the red data book, the species can be classified as rare, threatened, endangered and critically endangered, and finally it becomes extinct. So we have to conserve which are present in the critically endangered state or endangered state or threatened state, because once it gets extinct, there are no any means by which we can brought it back. So germplasm conservation in today's date is very important aspect. So what it includes actually, germplasm conservation includes the conservation of cultivated species, most importantly wild species, the local land races and breeding lines. So this is very important aspect that germplasm needs to be conserved in order to conserve the biodiversity on this earth, because conservation of biodiversity is very important to maintain the ecological or environmental balance. So let us discuss the types of germplasm conservation. Basically, there are two different types of germplasm conservation. First one is in situ conservation and second one is ex situ conservation. Let us see one by one. So what is in situ conservation? In situ conservation means on-site conservation of germplasm. Means what? The plants or the animals, they are conserved at their natural places. Means if the plant is present in the wild forest, it is conserved at that place only. So on-site conservation of germplasm or the preservation of germplasm in their natural habitat is termed as in situ conservation. In situ literally means in their natural habitat, therefore it is called as in situ conservation. So these germplasm conservations are conserved and protected. I told you earlier that they are conserved and protected in their natural environment. So what are the different types of in situ conservation method? First one is biosphere reserve. We take the example as a Nilgiri biosphere reserve or even other reserves which are present in the India. So these are the places, these are the protected places where the species of plants, forest, animals, they are conserved at their natural place. Then second is national park is there. For example, let us Jim Corbett National Park is there, Kanha National Park is there. So these are the protected environment, protected space, protected by the Forest Department of India, which conserve the species of different plants and animals, particularly the critically endangered or endangered or rare plants, or as well as the animals in their natural habitat. Third one is wildlife sanctuaries. For example, Karnala bird sanctuary is there. So these are also the areas, these are the protected areas where the plants or any living organism is conserved in their natural habitat. So these are the examples I told you earlier, that Jim Corbett National Park present in the Uttarakhand, which is an example of in situ conservation, Nilgiri Biosphere Reserve or Bharatpur Bird Sanctuary. So all these are the places where the species is conserved in its natural habitat. It means we are not going to bring them to other place for conservation. So that is exactly the next type of conservation. So which is next type of conservation? Next type of conservation is ex situ conservation. First we have seen in situ conservation, and this is the ex situ conservation method. So what is ex situ conservation method? It is termed as off-site conservation of germplasm. Off-site conservation of germplasm, or in other words, it can be defined as conservation of germplasm outside its natural habitat, is termed as ex situ conservation method. Means in situ we saw that we conserve the species into their natural habitat, but in case of ex situ conservation, we are not going to conserve them in natural habitat, we will bring them to some other places from their original habitat and try to conserve them. So in this method, genetic information of germplasm is preserved in the form of in vitro culture. What do you mean by in vitro? Vitreous literally means the glass. So in glass is present in the laboratories. So in vitro, this word means what? In laboratory conditions. In laboratory conditions means obviously outside its natural conditions. So in lab conditions, the genetic information present in the germplasm is conserved, and also the seeds are collected from their natural habitat and stored as gene bank or even the seed bank, which can be kept for the longer period of time. There is one example of Padma Shri Rahibai Popere. She has conserved more than 120 land races of the seeds, and for which she was awarded with the Padma Shri. So it's very big work, it's very great work to conserve the land races. So that type of conservation is called as ex situ conservation, because it is present outside their natural habitat. So what is the benefit of it? It forms the genetic information library for many endangered, rare, wild and commercially important species. And such genetic resources or germplasm are used for genetic improvement, biodiversity conservation, taxonomy and epidemiology. So this is the very important aspect why we should conserve, why we have to conserve this germplasm, because they can be used as a resource. I told you earlier that in hybridization technique, the two different varieties of the plants get crossed to produce the highly improved varieties, but for crossing the two different original varieties must be there. For crossing, original variety should be there, and that is provided through this conservation method. And I told you about biodiversity conservation, because it's very important aspect. We are living on this earth and we are just one part of this earth. compared to the, if we see other living organisms, and we are just one kind of living organisms, and other living organisms have equal right to be present on this earth, and for that biodiversity conservation is must, and it is very important, because to conserve, because we have to maintain the environmental balance. So, taxonomy is again the thing for which germplasms are required, epidemiology is again there. So, these seed banks and gene banks, which is a type of ex situ conservation method, they are the very valuable source for the breeders to develop the new and improved varieties. So, this is the example of seed bank. I have given the photograph of seed bank and gene bank. Gene bank is particularly nothing but the plant tissue culture technique, where you can conserve the part of the plant. It may be leaves, it may be stem, it may be a root part, and same plant part can be used as a germplasm, because plant has that property of totipotency. Totipotency means what? Ability of plant cell to generate an entire plant from the any plant part, and therefore any plant part can be treated as germplasm in case of plants. So, finally, I would like to thank you for your patient listening and attention. Thank you.